Hey guys, it's Morgan here with another TEAS bio lesson for you. This one is on understanding macromolecules. The lesson will start fairly basic and then work its way up to being a little bit more complex. So let's jump right in. So macromolecules are large molecules made up of smaller units called monomers. The monomers can join together through chemical reactions to form polymers, right? So poly. A polymer, right, that prefix poly means many. A polymer is a long chain-like structure made up of repeating units of monomers. Think of it like building with Lego, where monomers are individual Lego pieces and the polymer is the final structure you create by connecting all of those Lego pieces. There are two key processes involved in the formation and breakdown of polymers. One is dehydration synthesis, and the other is hydrolysis. Let's take each one in turn. Dehydration synthesis is the process by which monomers are joined together to form a polymer. Think of that word synthesis. Synthesis always means coming together. Water, as a molecule, is formed as a byproduct of dehydration synthesis, which is why you do that first word, dehydration. That's where that comes from. It's just that water is lost in the reaction that forms a polymer. During the reaction, a hydroxyl group, so remember that's like the hyphen OH, from one monomer combines with the hydrogen atom from another monomer. It creates a covalent bond, and this bond formation releases a water molecule. That's the dehydration part. This reaction occurs repeatedly, resulting in the formation of a polymer, with the elimination of water molecules each time a bond is formed. On the other hand, hydrolysis is the reverse process of dehydration synthesis. It involves breaking down the polymers, so deconstructing that Lego construction into their individual monomers, those individual Lego pieces, and it adds a water molecule in doing this. In hydrolysis, a water molecule is split into a hydroxyl group, an OH, and a hydrogen atom, an H, right? So that's one oxygen and two hydrogens. The hydroxyl group attaches to one monomer, while the hydrogen atom attaches to the adjacent monomer. This breaks that little covalent bond between them, and it separates the monomers. There are 4 key types of macromolecules that you're going to need to know to excel on your T7 bio exams. They are as follows: carbohydrates, proteins, nucleic acids, and lipids. Let's go through each type together. We'll start with carbohydrates. Carbohydrates, you may know them colloquially as sugars or starches. These are molecules composed of carbon, hydrogen, and oxygen atoms. They serve as the primary source of energy for all living organisms. You need carbohydrates, you need sugars to survive. The monomers of carbohydrates are called monosaccharides. Again, you see that prefix mono, meaning one. These simple sugars are joined together by dehydration synthesis to create larger structures, such as disaccharides or polysaccharides. Note that the prefix of each one of these words gives us a clue as to how many sugars are in each carbohydrate chain. So a monosaccharide has 1. A disaccharide, di means 2. Poly means many. So there are many sugars, 3 or more. Carbohydrates all have the same basic chemical formula. It's C, some number, H, 2 times that same number, O, and that same number as of the carbons. So the N subscript in this formula is a placeholder for how many of those atoms are in a particular carbohydrate chain. Each carbohydrate will have the same number of carbon and hydrogen atoms, and twice as many hydrogen atoms. So here are some examples that we're going to run through really quickly. The first is a sucrose, right? So if you were to count up the number of hydrogens, count up the number of oxygens that you see there, you will see kind of how many of each are present. Many monosaccharides linked together are called polysaccharides, like starch and cellulose. These are used for energy storage and structural support. Animals, including humans, store glucose, a monosaccharide, in the form of long glycogen, which is a polysaccharide, right? Those glycogen chains. Glycogen is formed through a process called glycogenesis, which is a dehydration synthesis. In animals, glycogen is stored in liver and muscle tissue. Anytime glucose is needed for cellular energy production, glycogen is hydrolyzed, right, broken down by the addition of water into glucose so that it can be used as energy. In plants, carbohydrates are stored primarily in the form of starch. Starch is a polysaccharide composed of glucose units and serves as an energy reserve in plants. It is stored in specialized plant structures such as roots, tubers, seeds, and fruits. That's where you find it. Plants also use a lot of carbohydrates called cellulose in their actual cell walls. Here's an image of polysaccharides. First, you see starch. We also have cellulose and then glycogen. The next type of macromolecule that you will need to know for your T7 exam is lipids. So lipids are a diverse group of molecules that include fats, oils, waxes, and steroids. The monomers of lipids are glycerol bonded to fatty acid chains. All lipids are hydrophobic. You're going to need to know that. That means that they do not dissolve in water. You've probably seen this before when you make a salad dressing, for example. The olive oil in the salad dressing doesn't mix well with the water or the vinegar that you put in there. You have to give it a good shake, right, before you pour it on your salad to make sure everything is mixed. This is because the oil will separate from the water unless it is forced to combine through your vigorous shaking. Lipids serve as a form of long-term energy storage. They also serve as insulation and also protection from organs, right? They are made up of fatty acids and glycerol. Fatty acids are long chains of carbon atoms with hydrogen atoms attached. Lipids can be saturated, meaning they contain only single bonds between carbon atoms, or unsaturated, meaning they contain one or more double bonds between the carbon atoms. Have you ever heard about the key differences between saturated and unsaturated fats with regard to your health? Let's look at how these different structures impact how each type of fat affects our bodies. So saturated fats have straight fatty acid chains and therefore can stack and compact easily one on top of the other. These are things like fatty meats and dairy. These fats tend to stay in a solid form and can cause things like fatty deposits in your blood vessels, which can lead to conditions like atherosclerosis. Saturated fats can also increase our LDL, or bad cholesterol. Experts believe that these fats inhibit special receptors in our livers that remove cholesterol from our bloods. Unsaturated fats, on the other hand, can be more beneficial to your health in a number of ways, including lowering your cholesterol levels, easing inflammation, and stabilizing heart rhythms. The bent structure of their fatty acid chains due to the double bonds are the main reason they're better for us than saturated fats. Lipids play an integral role in energy storage, insulation, cushioning, and the structure and function of cell membranes. One type of lipid that is crucial to the structure and function of the cell is the phospholipid. Phospholipids are the major component of cell membranes. They form a double layer called a phospholipid bilayer, with the hydrophilic, or the water-loving, heads facing outwards toward the watery environment, and the hydrophobic, or the water-fearing, tails facing inward. That creates a barrier that separates a cell from its surroundings. Understanding the different types of lipids and their functions help us appreciate their importance in maintaining health and function within all living organisms. The third type of macromolecule you'll need to know for your exam are proteins. Proteins are essential for the structure, function, and regulation of cells and tissues in our bodies. The monomers of proteins are called amino acids, right? There are 20 different amino acids. Every amino acid has the same basic structure. It has one central carbon atom attached to 4 different groups: a hydrogen atom, a carboxyl group, an amino group, and an R group. While the other 3 groups are always the same, the R group determines which amino acid it is. Each amino acid has different properties due to this side group, that R group. These varying side groups each give our proteins their various structures and properties. The link between amino acids is a covalent bond called a peptide bond, but it's just a covalent bond. Proteins have a specific three-dimensional shape that determines their function. They can act as enzymes, antibodies, transport molecules, and perform many other vital roles within the body. Proteins have different shapes and functions. For example, globular proteins are typically water-soluble, so that means they're hydrophilic. They perform functions such as enzyme catalyst, transport, and signaling. An example is hemoglobin, a globular protein that carries oxygen in red blood cells. The next is fibrous proteins. Fibrous proteins have long fiber-like shape, like their name would suggest. They are often involved in providing structural support through strength. Again, fibers, right? Thinking about fibers. Collagen, found in connective tissue like skin and tendons, is a fibrous protein. That's an example that helps maintain the structural integrity of these tissues. Finally, a very important protein are enzymes. They are remarkable and they play a crucial role in biochemical reactions. Enzymes lower activation energy required for a chemical reaction to occur. They make the reaction faster and more efficient. They are highly specific, acting on specific substrates like a puzzle and then sort of fit together. This specificity allows enzymes to carry out specific tasks in the body. Enzymes are not consumed or altered during a chemical reaction. This means they can be used repeatedly, making them essential for various and myriad biological processes. The final macromolecule that you'll need to be familiar with for your TEAS exam are nucleic acids, or is nucleic acid. Nucleic acids are composed of smaller units called nucleotides. These are the monomers. Each nucleotide contains a sugar molecule, a phosphate group, and a nitrogenous base. Again, this should sound familiar from our lessons on DNA and RNA. Nucleic acids are polymers made of nucleotides that contain carbon, hydrogen, oxygen, and nitrogen, all kind of the four key elements that are essential for life. They also contain phosphorus. Nucleic acids are responsible for storing and transmitting genetic information. The two main types of nucleic acids that we are familiar with, right, are DNA, which is deoxyribonucleic acid, and RNA, which is just ribonucleic acid. DNA is a double-stranded helix, again, this should sound familiar, that stores genetic information and carries the instructions for building and maintaining an organism. RNA is involved in protein synthesis. The sequence of the nucleotides in DNA and RNA determine the genetic code and the traits of a particular organism. DNA is made up of a double strand of nucleotides. Nucleotides are made up of a deoxyribose sugar, one of four nitrogenous bases, right? Adenine, thymine, guanine, or cytosine, and a phosphate group. Genes are made up of DNA. In humans, genes can contain a few hundred or more than a million DNA bases. RNA, on the other hand, consists of a single strand of nucleotides. The structure of RNA is similar to DNA, except it contains a ribose sugar molecule as opposed to a deoxyribose sugar molecule. Additionally, RNA has the same nitrogenous base as DNA, except it contains uracil instead of thymine. There are three main types of RNA. We have messenger RNA, transfer RNA, and ribosomal RNA. Key information about the differences between all four of these types of macromolecules, including what their function is, their structure, what the name of their monomer is. You want to keep that with multiple choice questions on this particular issue. And so the solution here is simply to make flashcards. All right, y'all. That concludes our lesson on macromolecules. Hey guys, it's Morgan here from Nurse Hub, ready to lead you in our biology course for the TEAS exam. We're going to begin with a tour of the cell. So buckle up and get ready for a very close review at all of the organelles and their functions that you're going to need to know to succeed on the bio section of the TEAS exam. All right, let's do this. Every living thing is made up from cells. Cells are essentially the building blocks of every living thing. They're the building blocks of life. They have some basic parts in common, although they come in different sizes and different shapes and they have different functions. There are some things that all cells have in common, and that's what we're going to focus on here for the TEAS 7 exam and what the exam will actually quiz you on. So there are three main parts to every cell. There's the cell membrane, the cytoplasm, and the nucleus. Let's first chat about the cell membrane. The cell membrane is the outer layer of the cell. It is made up of a thin, flexible layer of lipids and proteins, and it surrounds the cell and controls what can get in and out of the cell. It essentially acts as a gatekeeper, controlling the flow of nutrients and waste in and out of the cell. Cytoplasm is the next term you're going to need to know. Cytoplasm is a gel-like substance that fills the inside of a cell. It is made up of water, salts, and various types of molecules, including proteins, enzymes, and nutrients. The cytoplasm is where many of the cell's metabolic reactions take place, such as the production of energy, or ATP, through cellular respiration. The next term that I would like you to focus on is the nucleus. This is the control center of the cell. It's usually located near the center of the cell, though not always, and it is surrounded by a double membrane called the nuclear envelope. Inside the nucleus is some of the most important stuff to do with a cell. It includes the cell's DNA, which contains the genetic information the cell needs to function and reproduce. You'll learn more about DNA and genetic material later in this course. The nucleus also contains the nucleolus, which is responsible for making ribosomes, the cell's protein-making factories. All right, so let's focus in on the cytoplasm. The cytoplasm is where we find all of the organelles. Organelles are specialized structures that perform essential functions on behalf of the cell. Think of them as the cell's organs, right? These are organelles. They work within the cell to make it work. There are several organelles that you're going to need to know in order to be successful on the biology section of the TEAS 7 exam. The first is mitochondria. Mitochondria are the organelles responsible for producing energy within the cell. They are often called the powerhouses of the cell. And we'll spend some time focusing on them in just a moment in our next video and what they do and how they produce that power. The next is the endoplasmic reticulum, or the ER. This is a network of membranes that help transport proteins and other materials throughout the cell. They're super important, and we'll also have another lesson on that. The next is the Golgi Apparatus. This is another membrane-bound organelle, so it is kind of stuck in position within the cell by a membrane. It helps to process and package proteins for transport outside of the cell. Lysosomes are the next organelle that you'll need to know. These are small organelles. They contain enzymes that break down waste and other cellular debris. Another key structure of a cell is the cytoskeleton. The cytoskeleton is a network of protein fibers that help give the cell its shape and provide support structurally for the cell. All right, guys, those are sort of some of the major organelles that you're going to need to know for the TEAS 7 exam. Join me as we proceed through all of the subsequent lessons, which will focus on each one of these organelles in particular, giving you all the information you need in order to be successful on the TEAS exam bio section. Until next time. Hey guys. All right, so let's jump right in as we continue our lessons on the tour of the cell. In this lesson, we're going to discuss the importance of mitochondria within the cell. As you know, mitochondria are the powerhouses of the cell. In this video, we'll be taking a closer look at the structure, function, and role of mitochondria in the cell. As an overview, mitochondria are small, semi-autonomous organelles found in the cytoplasm of eukaryotic cells. They are responsible for producing the majority of the cell's energy in the form of ATP through the process of cellular respiration. Just so you know, ATP stands for adenosine triphosphate. That's the energy that is used by cells. We're going to go into more depth on that later, so don't freak out yet, but that's what ATP is. Mitochondria have a unique double membrane system with an outer membrane and an inner membrane. The inner membrane is folded into cristae, which greatly increase the surface area for energy production. It also contains specific enzymes that are involved in the production of ATP. The space between the two membranes is called the intermembrane space, while the space within the inner membrane is called the matrix. This is where the process of chemiosmosis occurs. During chemiosmosis, hydrogen ions are pumped across the inner membrane of the mitochondria, creating a proton gradient. This proton gradient is then used by the enzyme ATP synthase to generate ATP, which is the main source of energy for the cell. This is why the mitochondria is called the powerhouse. It's literally keeping the lights on in the cell. Mitochondria have their own genetic material separate from the cell's nuclear DNA. They also have their own ribosomes, which are responsible for synthesizing proteins. The proteins synthesized within the mitochondria are mostly involved in the production of energy. Interestingly, mitochondria play a key role in cell death, known as apoptosis. Apoptosis is a programmed process that occurs in cells in response to certain signals, such as DNA damage or viral infection. When a cell receives these signals, the mitochondria release a group of proteins called cytochrome C, which triggers a cascade of events that lead to the death of the cell. One instance of apoptosis that most of us experience is the programmed cell death of skin cells that act as webbing between our fingers and toes while we're in utero. You may not know this, but when we are in utero, we have webbing between our fingers and toes. But by the time we're born, that webbing is gone. That's because of apoptosis, coded cell death, telling the cells, you can leave now. You're not needed. In summary, mitochondria are the powerhouses of the cell, with a unique double membrane structure. They are semi-autonomous due to the presence of their own genetic material, and ribosomes. Those ribosomes are responsible for protein synthesis and energy production. Not only that, but mitochondria play a key role in programmed cell death. Thank you for watching. We hope you now have a better understanding of the importance of mitochondria in the functioning of cells. Hey guys, all right guys, welcome to the lesson on the Golgi Apparatus. The Golgi Apparatus is an organelle found in eukaryotic cells that plays a key role in the modification, sorting, and transport of proteins and lipids. It is composed of a series of flattened, stacked cisternae, which are membrane-bound structures organized into three regions: the cis Golgi, the medial Golgi, and the trans Golgi. The cis Golgi is the region closest to the endoplasmic reticulum, or ER, and is responsible for receiving newly synthesized proteins and lipids from the ER. It is characterized by its cis face, which is the side of the cisternae facing the ER. This is where incoming proteins and lipids enter the Golgi. The medial Golgi is the region between the cis Golgi and the trans Golgi, and is responsible for further modification of protein and lipids. It is characterized by the presence of specific enzymes that add carbohydrate and lipid modifications to the proteins and lipids. The trans Golgi is the furthest region from the endoplasmic reticulum, and it is responsible for sorting and directing the modified proteins and lipids to their final destinations. It is characterized by its trans face, which is the side of the cisternae facing away from the endoplasmic reticulum. The trans face is where the modified proteins and lipids are packaged into vesicles and transported to their final destinations, such as the plasma membrane for secretion or lysosomes for degradation. The Golgi apparatus is an essential organelle that plays a crucial role in the modification, sorting, and transport of proteins and lipids in eukaryotic cells. Hey guys, welcome to our video on lysosomes. These powerful little organelles play such an important role in maintaining a stable and balanced environment within the cell. In this lesson, we're going to explore the structure and function of lysosomes and how they help maintain the cell's health and what can happen if they're damaged. Let's jump in. Our cells are often compared to tiny cities, right? Cities need to get rid of waste to function properly. Just like how cities have recycling centers to process waste, cells have lysosomes to degrade and recycle old or damaged materials. Lysosomes are organelles that contain special enzymes called hydrolases, which break down complex molecules into simpler components. This process is known as autophagy. Autophagy is a cellular process in which cells degrade and recycle their own damaged or unnecessary parts. It involves the formation of phagosomes, which are double membrane vesicles that fuse with lysosomes to degrade their contents. Autophagy is an important process for maintaining cellular homeostasis and supporting normal cellular functions, as well as development, immunity, and stress responses. Let's look at an animation of this process. The job of a lysosome is to digest anything that isn't needed or wanted inside the cell. After something slated for digestion is encased in a phagosome, the lysosomes will fuse it with the vesicle. In the case of old or damaged cell parts, the lysosome may fuse with a particular organelle that needs to be broken down and recycled. Once the lysosome has fused with either a vesicle or an organelle, the acidic inside of the lysosome activates the hydrolases to break down and recycle the cellular waste, damaged organelles, and even bacteria. Lysosomes work in consort with other organelles, such as the Golgi apparatus, to ensure efficient processing and complete elimination of cellular waste. This is crucial for maintaining cellular homeostasis, which is essential for the proper functioning and the health of the cell. These organelles are so essential to the proper functioning of the cell that lysosomal damage can lead to the buildup of harmful materials within the cell, causing cellular stress and potentially triggering programmed cell death or apoptosis. Hey guys, all right guys, welcome to our lesson on the cytoskeleton. The cytoskeleton is not an organelle, but it is such an important part of the cell that it is included on our tour of the cell. The cytoskeleton provides kind of the shape and form for a cell. It is made of a complex network of protein fibers that support with obviously shape, structure, but also mechanical functionality of the cell. One of the key functions of the cytoskeleton is to facilitate the movement of materials within the cell. The cytoskeleton is composed of three main types of fibers. You'll need to know these for the TEAS exam. The first is microfilaments. The second are intermediate fibers, and the last are microtubules. We're going to go through each one of those types of fibers so that you really understand the features and what distinguish them between one another, or from one another. Microfilaments, or actin filaments, they will be often referred to as actin filaments interchangeably in different study materials or course prep, so keep that in mind. Actin filaments and microfilaments are the same, and those terms are interchangeable. They are thin, flexible fibers made of the protein actin, hence the name actin filaments. They create a constantly changing network in cells that is essential for many functions. Microfilaments help the cell move by forming temporary extensions called pseudopodia, fake feet, right? They are also important for moving organelles and other materials around the cell, which is called cytoplasmic streaming. Additionally, microfilaments can bundle together to help the cell keep its shape. Think of them like the flexible pipes that run through a building, allowing water and other materials to flow where they need to go. Microfilaments also play a very important role during cellular replication and division, most notably during cytokinesis, when the cell splits into two daughter cells, right? So during that process, microfilaments contract, they form a furrow in the middle of the parent cell, and the microfilaments form a ring that gets smaller as they contract. The cytoplasm is pinched until the original cell is split into two daughter cells. You'll learn more about this process in our lesson on mitosis later in this course. Let's talk about intermediate fibers. So intermediate fibers are thicker and more rigid than microfilaments, but they're not as thick as microtubules. That's an important distinction, and that will come up on the exam, or often comes up on the exam. They provide the cell with mechanical strength and support. They are made up of a variety of proteins, including keratin, desmin, and vimentin. While intermediate fibers are not directly involved in the movement of materials within the cell, they provide a stable framework that allows for the proper organization of other cytoskeletal elements. Microtubules, final type of fiber, are long tubular structures that play a critical role in maintaining the shape of and organization of cells. They are a key component of the cytoskeleton, which is a network of protein fibers that provide the structural support that helps with cell division, cell movement, and cell intracell communication. One important aspect of microtubules is that they are made up of dimers, which are pairs of protein subunits called alpha-tubulin and beta-tubulin. These dimers are arranged in a head-to-tail fashion to form long chains, which then assemble into a cylindrical shape of a microtubule. The formation of dimers is important for microtubule stability and function. For example, when a microtubule is assembled, the dimers are positioned in a specific way that allows for the microtubule to grow and shrink in a controlled manner, right? The dimer formation allows proteins to travel along the microtubules. Two of these traveling proteins are motor proteins called kinesins and dyneins. Kinesins are motor proteins that move or walk toward the positive end of a microtubule, while dyneins travel toward the negative end. These proteins typically carry vesicles to their destinations. This dynamic behavior is important for many cellular processes, such as cell division, cell movement, and cell organization. In addition, the binding of certain drugs or proteins to microtubules can disrupt the formation of dimers or cause them to become misaligned. This can alter the stability and the function of the microtubule. This has important implications for medical research, as drugs that target microtubules can be used to treat diseases such as cancer or Alzheimer's. Microtubules are also major components of mitotic spindle fibers, which separate sister chromatids during mitosis. One end of the spindle fibers attaches to the center of each sister chromatid, while the other end of the fiber attaches to one of the two centrosomes at the end of each cell. The spindle fibers begin to shorten and pull the sister chromatids apart to opposite ends of the cell. Again, you'll learn more about this process in our mitosis lesson later on in this course. As you can tell, the cytoskeleton plays a critical role in the structure and function of a cell. Hey guys! All right, welcome to our lesson on cilia, flagella, and centrioles. We're focusing on these as part of our tour of the cell because they're such important structures for the functionality of a cell. Cilia and flagella are microscopic hair-like structures that protrude from the surface of certain cells. They are composed of microtubules and play an important role for cellular movement, sensory perception, and fluid movement. So we're going to focus now on just flagella and cilia to begin with. So flagella are typically longer and fewer in number than cilia. They are used for locomotion, meaning movement right throughout the cell. They can be found in various organisms, such as bacteria, algae, and sperm cells even. For example, in bacteria, flagella rotate like propellers to propel the cell through liquid environments, while in sperm, the flagellum propels the sperm cell, toward the egg. Cilia, on the other hand, are shorter and more numerous than flagella. They can be found in many types of cells, including those in the respiratory tract, oviducts, and the brain. In the respiratory tract, cilia help move mucus and foreign particles out of the lungs, while in the oviducts, cilia help move the egg toward the uterus. In the brain, cilia play a role in sensing and transmitting signals. Overall, the function of cilia and flagella are critical for many biological processes, including cell movement, sensing things in the environment, and fluid transport. Without these structures, many organisms would not be able to survive and carry out their essential functions. The centrosome is a crucial organelle that regulates the organization and growth of microtubules in the cell. Positioned near the nucleus, it serves as the microtubule organizing center. The centrosome is composed of two cylindrical centrioles, which have a unique pattern of microtubule arrangement. When a cell divides, the centrosome duplicates and moves to opposite parts of the cell, where it plays a crucial role in organizing the microtubules that make up the mitotic spindle. All right, now let's discuss centrioles. Centrioles are small cylindrical structures found in most eukaryotic cells. They play an important role in cell division and organization of microtubules in the cytoplasm. During cell division, centrioles replicate, duplicate, and move to opposite ends of the cell, where they help organize microtubules into spindle fibers. These spindle fibers attach to the chromosomes and pull them apart during cell division, This ensures that each new cell receives the correct number of chromosomes. Centrioles also play a role in organizing microtubules in the cytoplasm. This is important for maintaining the cell's shape and supporting the movement of cellular structures. The microtubules that originate from the centrioles can form into cilia and flagella. In addition to their role in cellular division and organization of microtubules, centrioles have also been implicated in a range of cellular processes, including cell signaling and regulation of DNA repair. While centrioles are not essential for cell survival, they are critical for proper cell division and the function in many types of cells. Defects in centriole function have been associated with a variety of human diseases, including cancers and ciliopathies. Hey guys, all right, the next part of the cell that we're going to focus on is the cell membrane. It's also known as the plasma membrane. Those terms will be used interchangeably, so be on the lookout. The cell membrane is a thin, flexible barrier that surrounds and encloses the contents of a cell. Its most important function is working to maintain the cell's internal environment, right? So without that barrier, the cell would have no internal environment. So the cell membrane keeps everything together in a little pouch and forming like the physical object and structure that we can identify as a discrete cell separate from others. One way the cell membrane does its job is by controlling the passage of substances into and out of the cell. The membrane is selectively permeable, meaning it allows certain molecules, such as oxygen, carbon dioxide, and small hydrophobic molecules to freely diffuse across it, right, entering the cell or exiting the cell, while restricting the passage of larger, polar, or charged molecules. This selective permeability is due to the structure of the membrane, which is composed of a bilayer of phospholipid molecules, also known as just a phospholipid bilayer. Phospholipids are amphipathic, meaning they have both hydrophobic and hydrophilic regions. This allows them to form a stable bilayer in an aqueous environment. The hydrophobic tails of the phospholipids face inward, while the hydrophilic heads face outward. This creates a barrier that separates the inside of the cell from the outside environment. Another important function of the cell membrane is to facilitate cell-to-cell communication and signaling. The membrane contains various receptors and proteins that allow the cell to detect and respond to external stimuli, such as hormones, neurotransmitters, and pathogens. These signals can trigger a cascade of molecular events within the cell that ultimately result in changes in gene expression, cell growth, differentiation, or apoptosis. In addition, the membrane can form junctions with nearby cells, which allows the cells to adhere to one another and coordinate their activities, as is the case in tissues and organs, right? That's just tons of cells formed together to perform a particular function, a large function. Finally, the cell membrane also plays a role in maintaining the structural integrity of the cell. The membrane is anchored to the cytoskeleton, a network of protein filaments that provide support and shape to the cell. The membrane also contains various lipids and proteins that contribute to its fluidity and stability. This allows it to withstand physical stress and changes in temperature or pressure. Additionally, the membrane can actively transport molecules across the membrane, using energy derived from ATP hydrolysis or ion gradients, which is critical for various cellular processes such as nutrient uptake, waste removal, or signaling. Overall, the cell membrane is a highly dynamic and complex structure that performs numerous functions in maintaining the viability and functionality of the cell. I hope you've gained a better understanding of how important the cell membrane is to a cell's health and function. Hello everyone, welcome back. Morgan here to go over mitosis and meiosis with you. So in this lesson, we're going to cover both mitosis and meiosis, cell division, and compare and contrast similarities and differences, which is information you're going to need for the TEAS 7 exam section on biology. All right, so let's jump right in. So mitosis is the process by which a cell divides and produces two identical daughter cells. It is a critical process for growth and repair in multicellular organisms like humans. The process of mitosis is divided into 4 different stages. Those stages are prophase, metaphase, anaphase, and telophase. So let's look at each one of those stages in detail. Interphase. So interphase is the stage of the cell cycle that occurs before mitosis actually begins. Interphase is important because it is the time when the cell is preparing to divide into 2 daughter cells. During interphase, the cell increases in size, it produces additional organelles and proteins, so it's duplicating all of these things and duplicates all of its DNA as well. The cell then has 2 copies of its DNA going into mitosis that can be separated into 2 sister chromatids as the DNA condenses and provide the genetic code for those 2 identical daughter cells. The next stage is called prophase. Prophase is the actual first stage of mitosis. It is where the cell begins the process of physically dividing into 2 daughter cells. At the beginning of prophase, the chromatin, which is a combination of DNA and proteins, starts to condense. The condensing causes the chromatin to coil and compact, resulting in the formation of distinct structures called chromosomes. Chromosomes are easier to manage and distribute during cell division, right? They're like a package of very critical DNA information. Another process is happening in prophase parallel to that, which is the breakdown of the nuclear envelope. The nucleus of the cell is surrounded by a membrane, which we've discussed. That membrane is also sometimes called the nuclear envelope. It separates the nucleus from the rest of the cell and allows the nucleus to have its own distinct identity separate from the cell. This breakdown allows the chromosomes to be exposed to the cytoplasm of the cell. The next step is the formation of the mitotic spindle. Fun word, mitotic. The mitotic spindle is a structure composed of microtubules that helps in the movement and separation of chromosomes during cell division. In prophase, 2 centrosomes, which are small organelles located near the nucleus, move to opposite ends of the cell. As the centrosomes move, they generate spindle fibers that extend from each centrosome toward the center of the cell. These little spindle fibers form a complex network known as the mitotic spindle. As the mitotic spindle develops, it interacts with the chromosomes. Specialized protein structures called kinetochores form at the centromere region of each chromosome. It's that kind of the intersection of where you kind of see like the X, it's right there. It serves as an attachment point for those spindle fibers. Then those spindle fibers attach to the kinetochores and help align and position the chromosomes in the cell. So kind of like trying to align buoys or something, right? You kind of imagine physical structures in a fluid environment needing to be kind of tethered to one another and positioned using each other as anchor points so that this process can occur. And when the division occurs, each cell that is created has exactly the same as what the other one has. Now let's go over metaphase. That's the next stage of cell mitosis, where chromosomes line up in the center of the cell and they get ready to divide. It's like a line dance, right? They're all getting lined up, ready to do their thing. In prophase, the chromosomes begin moving toward the center of the cell. During metaphase, the spindle fibers continue to exert tension on the chromosomes, aligning them along the metaphase plate at the center of the cell, right? That alignment ensures that each chromosome is properly positioned for the upcoming separation. The metaphase plate is an imaginary plane that runs through the center of the cell and is perpendicular to the spindle fibers. So the plate, the metaphase plate, is really just sort of a figure of speech. It's a concept that allows our brains to wrap around the concept of what's happening, but there is no physical structure that will be labeled metaphase plate, for example, on your exam. The chromosomes line up single file along this plate or plane. The alignment is essential for ensuring that each daughter cell receives an equal and complete set of chromosomes during cell division. Before moving into anaphase, the cell completes a quick checkpoint to make sure that all of the chromosomes are lined up correctly, that all of them are connected to the spindle fibers by their kinetochores. And then during metaphase, the chromosomes are at their most condensed and visible state. They appear as distinct structures with two sister chromatids held together by a centromere. The next stage is anaphase. In anaphase, the spindle fibers shorten and pull the sister chromatids, those two identical parts of the chromosome, apart and toward the opposite poles of the cell. At the start of anaphase, the connections holding the sister chromatids together at the centromeres dissolve. This allows the sister chromatids to separate and become individual little chromosomes. Each chromosome now consists of a single chromatid. Once the sister chromatids have separated, they move toward the opposite poles of the cell, again, creating some space for the drama that's about to happen as those cells go ahead and separate. This movement is facilitated by the shortening of those spindle fibers, kind of thinking about those spindle fibers as ropes being pulled in and pulling those chromosomes or those chromatids toward one end or the other of the cell. As those spindle fibers contract, they pull the chromosomes in the direction of the centrosomes located at the opposite poles of the cell. The separated chromosomes move in a V-shaped configuration toward the poles. So it's kind of like as they come toward the pole of the cell. As those sister chromatids are moving toward the opposite poles, the spindle fibers not attached to chromosomes elongate, okay? The elongation of the unattached spindle fibers helps to further separate the poles of the cell, widening the distance between them. Okay, so it's sort of expanding the cell out, widening the space so that when that separation happens, again, this is all about facilitating and creating ease, and also ensuring parity between both of those new cells that are created. While anaphase is primarily focused on the separation and movement of chromosomes, it also marks the beginning of cytokinesis, the division of the cell's cytoplasm. In animal cells, a contractile ring composed of actin and myosin proteins form the center of the cell. The ring contracts, leading to the formation of a cleavage furrow that eventually pinches the cell into two daughter cells. Next is telophase. During telophase, the spindle fibers disappear. The nuclear envelope reforms around each set of chromosomes. The chromosomes begin to uncoil, and the cell begins to divide, creating those two identical daughter cells that we talked about earlier. In telophase, the separated chromosomes, which migrated to opposite poles during anaphase, reach their respective poles. At this point, two distinct groups of chromosomes are present at each pole of the cell. As telophase progresses, the chromosomes begin to decondense or uncoil. They transition from their highly condensed state, right, those sort of familiar chromosome shapes, back into their extended form of chromatin. Decondensation allows the genetic material to become accessible for gene expression and other cellular processes. Telophase is characterized by the reformation of the nuclear envelope around each set of chromosomes. The nuclear envelope is made up of two phospholipid bilayers, which you will remember from our prior lesson on the nucleus, and it separates the genetic material from the rest of the cell. The nuclear envelope reforms by the fusion of small vesicles derived from the endoplasmic reticulum, creating two distinct nuclei within the cell. Within each newly forming nucleus, the nucleolus, which is responsible for producing ribosomes, reappears. The nucleolus is composed of RNA, proteins, and DNA segments, and it plays a vital role in protein synthesis. Telophase also marks the completion of cytokinesis, which is the process of dividing the cell's cytoplasm into the two daughter cells. After mitosis is complete, the two new daughter cells will continue to grow and carry out their functions. identical to the parent cell that produced them. Mitosis is important because it allows the cells to divide and produce new cells for growth and repair. And it also plays a crucial role in asexual reproduction in some organisms. All right, everyone, that concludes our lesson on mitosis and all of the different phases. All right, hey guys, it's Morgan here with another lesson for your biology section of the TEAS 7 exam. This lesson is going to be on meiosis. So meiosis is a type of cell division like mitosis, but this one results in the creation of 4 daughter cells that each have half the number of chromosomes of the parent cell. Whereas in mitosis, you're creating 2 daughter cells that are identical. In meiosis, you're creating 4 daughter cells, and each of them have half the number of chromosomes as their parent cell. This occurs in the production of gametes and plant spores. So when we say gametes, obviously what we mean are the initial phase of like a human embryo. We're going to go through all the phases of meiosis, and then you can move through the lesson as you compare and contrast the differences in the phases and the processes involved in meiosis and mitosis. So let's jump right in. The first phase of meiosis is called interphase. During that time, the cell actually enlarges and it carries out its normal functions, as well as duplicates its DNA. This DNA duplication ensures that each chromosome is composed of 2 identical sister chromatids. It's very similar to mitosis. There are kind of 2 tracks of meiosis. And so often the first one is called meiosis 1 and the second is called meiosis 2. Let's focus now on meiosis 1. The first stage of meiosis 1 is called prophase 1. At this stage, the cell prepares for division. The nuclear membrane starts to break down and the chromosomes condense and become visible. The most significant event in prophase 1 is homologous chromosome pairing. Homologous chromosomes, which are a pair of similar chromosomes, one from each parent, come together and form a structure called a tetrad. During this pairing, sections of the chromosomes may exchange genetic material through a process known as crossing over. This genetic exchange promotes genetic diversity within families and within larger populations of a particular species. Next is metaphase 1. The homologous pairs of chromosomes line up along the center of the cell, called the metaphase plate, and become attached to spindle fibers. Again, you'll remember from the lesson on mitosis, those spindle fibers are kind of like ropes. They exercise some tension on the parts of the chromosomes to pull them and maneuver those parts to the correct location they need to be in the cell. The alignment of the chromosomes is random, leading to independent assortment, which further increases genetic diversity. The next stage is called anaphase 1. In anaphase 1, the homologous chromosomes separate and are pulled to the opposite pole by the spindle fibers. The sister chromatids remain attached to each other at their centromeres. And now the final stage of meiosis 1 is called telophase 1. During telophase 1, the separated homologous chromosomes reach the opposite poles of the cell. The nuclear membrane reforms around that set of chromosomes, and the cell undergoes cytokinesis, resulting in those 2 daughter cells. Each daughter cell, though, only has 1 chromosome from each homologous pair, and each chromosome consists of 2 sister chromatids. Next is meiosis 2. Meiosis 2 begins with prophase 2. So the daughter cells that were created in meiosis 1 enter a brief interphase where no DNA replication occurs. The chromosomes recondense, the nuclear membrane begins to break down, and then you enter metaphase 2. The chromosomes align individually along the metaphase plate attached to spindle fibers. And then in anaphase 2, the centromeres divide and separate the sister chromatids. The spindle fibers pull the chromatids to the opposite ends of the cell. And then you have telophase 2, where the chromosomes reach the poles of the cell, nuclear membranes form around each set of chromosomes, cytokinesis occurs, resulting in the formation of 4 haploid daughter cells. These cells contain half the number of chromosomes as the original cell, and each chromosome consists of only 1 chromatid. Okay, so it's different than mitosis in that way. The 4 daughter cells produced at the end of meiosis 2 are genetically unique due to the random assortment of chromosomes
in Meiosis 1, the exchange of genetic material that also occurred during Meiosis 1. And these cells serve as gametes, sperm or egg cells, and can combine during fertilization to create a new individual with a diverse genetic makeup. All right, guys, that's our video lesson on meiosis. Hey guys, welcome to our video lesson on comparing DNA, chromosomes, and genes. Let's jump right in. All of our characteristics, from physical traits to unseen bodily processes, are coded for in our genetic makeup. You'll probably recognize the terms we're about to cover from your biology classes of years past. And it's important to understand the role of each level of genetic material and how they relate to each other. On the most basic level, DNA is organized into genes, which are further organized into chromosomes. It's important to understand both their connections and how they are different from one another. We'll start with the most basic level of genetic material everyone is familiar with that exists in our cells, called DNA. If you've watched any true crime show, you've heard this term over and over and over again. So let's really dive in and figure out, what is DNA? DNA, which is an acronym for deoxyribonucleic acid, is a molecule that contains the genetic instructions for the development and functioning of all living organisms. It is often referred to as the building block of life because it carries the information that determines the characteristics and traits of an individual. It also carries the directions our cells need to perform all of their functions and produce the right proteins that our bodies need. Let's first talk about nucleotides. DNA is found in the nucleus of cells and is made up of smaller units called nucleotides. Each nucleotide contains a sugar molecule, a phosphate group, and a nitrogenous base. There are 4 types of nitrogenous bases in DNA. You may have heard them before. These names may ring a bell, again, from those bio classes from years past. The first nucleotide is adenine. It has an acronym, which is simply the first letter of the word, A. The next is thymine. The acronym for thymine is a capital T. The next is cytosine. The acronym for that is simply a C. And guanine, which is represented by a capital G. You'll notice that these 4 bases have different structures. This is important to note as it affects how they bond to one another. The bonds between 2 strands of DNA are very, very specific. Adenine always bonds with thymine and is held together with 2 hydrogen bonds. Guanine always bonds with cytosine and is held together by 3 hydrogen bonds. These bases couldn't bond in other ways because the uneven hydrogen bonding sites would make the structure very unstable. Adenine could not bond with cytosine because adenine does not have a 3rd place in its structure for another hydrogen bond. The order and arrangement of these bases along the DNA molecule creates a specific genetic code. The sequence of the bases in DNA determines the specific instructions for building and maintaining an organism. We often think about our genes as the characteristics that make each of us unique, but those genes are only a small set of our DNA and genetic code. The majority of our genetic code is instructions for our cells to function properly. Here's a quick kind of nurse hub tip, which is that DNA is inherited from our parents. That's why we share certain physical traits with our family members. We'll cover this part of inheritance in more detail in our lesson on Mendel's laws of inheritance. You may be wondering, how does DNA actually get into the nucleus of our cells? So you may recall from our lesson on mitosis that organisms grow and develop through a process called cell division. Before cell division takes place, DNA has to replicate. Remember the identical sister chromatids that separate and then they form 2 new cells? Well, that's exactly what I'm talking about. DNA replication ensures that each new cell receives an exact copy of the instructions, right, of the genetic information that tell it what to do. We will cover the process of DNA replication in more detail in the next lesson on protein synthesis. Mutations, or changes in DNA code, can occur during the replication process. Mutations happen spontaneously or due to external factors like radiation or certain chemicals. Some mutations can have no significant impact on our cells. They occur, they are harmless, and our cells move on and are still able to conduct their functions properly. Other mutations can cause genetic disorders or lead to variations in traits within our population. So let's zoom out a little bit and talk about genes. So genes are specific segments of DNA, of the DNA code, right, the double helix, that carry instructions for creating specific proteins which are essential for various biologic functions. Remember that DNA is one long molecule. There are many proteins coded within a single strand of DNA. So there has to be a way for the cell machinery to locate a particular section of code that instructs it on how to do or make a specific protein. Genes are the answer to this problem. If the cell's machinery is responsible for making proteins, it reads the strand of DNA from beginning to end, it wouldn't be able to make sense of it. The machinery within a cell cannot read multiple genes at one time. Each one of the sets of instructions needs to be sort of separated out like a filing cabinet. Each gene is a specific segment of DNA that only codes for one specific protein. We'll have 2 copies of each gene in our DNA. We get 1 copy from our biological mother and 1 copy from our biological father. You will learn more about how and why certain versions of genes are expressed in each individual when we talk about dominant and recessive traits in our lesson on Mendel's laws of inheritance. So if we zoom out a little bit more, we have chromosomes. Chromosomes are thread-like structures that are found within the nucleus of the cell, sort of an organizational structure. They are made up of DNA along with the proteins that help organize and support the DNA itself. Humans typically have 46 chromosomes, which exist in 23 pairs, with 1 set inherited from each parent. Each chromosome contains numerous segments called genes. Remember those from just a moment ago? Genes are specific stretches of DNA, right? From this adenine and thymine pair to this cytosine and guanine pair. That's a gene, right? Those genes carry the instructions for creating proteins, which are essential for various biological functions. These proteins determine the traits and characteristics of an organism, such as eye color, height, or blood type. Chromosomes carry specific genes. So the traits we inherit from our biological parents are organized on those same chromosomes. Recent research has allowed scientists to locate where certain genes are on particular chromosomes. Chromosomes also play a role in genetic disorders. Genetic disorders are caused by abnormalities or mutations in specific genes located on particular chromosomes. These mutations can lead to a disruption in protein production or cellular function, resulting in various health conditions or inherited diseases. So very quickly, let's review the relationship between the 3 basic levels of genetic material organization. Remember that from largest to smallest, genetic material exists 1 as DNA. Then there are segments of DNA that are called genes. And then all of this is wrapped up into chromosomes that look kind of like X's, right? And there are 23 pairs. We get half from our mom, our biological mother, and half from our biological father. To visualize this relationship, think of DNA as a long string, like a necklace, made up of different beads. The beads represent genes, which are specific segments of the DNA. Now imagine that you take that necklace and wind it into small balls. Those balls represent proteins. And when the DNA is wrapped around the proteins, it forms the structure of a chromosome. All right, guys, that's our lesson on DNA, genes, and chromosomes. Hey guys, welcome to our lesson on RNA. Now, before we get into the process of building proteins from DNA sequences, aka genes, we need to review the other type of genetic material, which you may recall from grade school. It's called RNA. RNA stands for ribonucleic acid. It is the other type of nucleic acid in our cells and has some key differences in structure that allow it to do its specific job that's different from DNA. RNA is the key to our ability to use our DNA to manufacture proteins necessary for our cells to function. It takes the information from our DNA and carries it over to the ribosomes, which manufacture the proteins on the genes that contain certain sequences of DNA. RNA structurally is similar to DNA, but there are some key differences that I really want to point out to you. The first is that DNA is a double-stranded molecule. It's known as sort of like a double helix, right? The two halves are zipped together, they're connected. Whereas with RNA, it is one strand of nucleotides only. Another key difference between DNA and RNA is the sugar base that is involved in the creation of the structure itself. So with RNA, it is a ribose sugar, whereas with DNA, it is a deoxyribose sugar. That's where they get their names from. It's based on the discrepancy between the types of sugar that's involved in their formation. The final key difference between DNA and RNA is the nitrogenous bases. So DNA uses adenine, thymine, cytosine, and guanine, whereas RNA uses adenine, cytosine, guanine, but in place of thymine, it uses uracil. Okay, and that's represented with a U. This is important for the ribosomes in reading the genetic code that creates the proteins. We've already covered how the base pairs match in DNA. The only difference in base pair matching in RNA is that DNA pairs with uracil instead of thymine in RNA. We'll talk more about base pairing in RNA in our protein synthesis lesson that follows this one. Finally, one key difference between DNA and RNA is that they have two very different functions. So we know that DNA carries specific genetic information needed to provide instructions to our cells. This is also the genetic information that is passed from our parents to their offspring. The main function of RNA, on the other hand, is to transfer genetic information from the DNA to the ribosomes so that the proteins can actually be made. So we've outlined the key differences between DNA and RNA in this table here. You can easily take notes about the differences and study and make flashcards for yourself. So the first is in their structure. As I previously outlined, DNA is known as a double helix. It is two-stranded, contains two complementary strands that are connected with hydrogen bonds between the nucleotide pairs. The nucleotides in DNA are adenine, guanine, cytosine, and thymine. RNA, on the other hand, is typically single-stranded, although it can fold back on itself to form like more complex structures. It contains ribose sugar instead of deoxyribose sugar that is found in DNA, and the four bases in RNA are adenine, guanine, cytosine, and uracil. We also know that the base pairing is different, so those nucleotide pairings. In DNA, we know adenine and thymine pair together. We also know that cytosine and guanine pair together. In RNA, adenine pairs with uracil, not thymine, and cytosine still pairs with guanine. And then finally, the function. So DNA carries the genetic information that determines our traits and characteristics. It serves as the long-term storage of genetic information and is primarily involved in the replication and inheritance of genetic material. RNA, on the other hand, plays a crucial role in protein synthesis. It acts as an intermediary between the DNA and RNA, transmitting information from the DNA itself to the ribosomes so that the ribosomes can create proteins that are then synthesized and involved in various other cellular processes, such as gene regulation and catalysts. So for your TEAS exam, there are three main types of RNA that you need to keep track of. The first is messenger RNA, or mRNA. mRNA carries genetic instructions from the DNA to the ribosomes. It acts as a template or a messenger by transcribing DNA code and then running it over to the ribosomes where the protein synthesis actually occurs. The next type of RNA that you need to keep track of is transfer RNA, or tRNA. tRNA helps in the translation process by bringing amino acids, the building blocks of proteins, to the ribosomes itself. Each tRNA molecule is specific to a particular amino acid, and it ensures that the correct amino acids are brought to the ribosomes according to the mRNA instructions. The third type of RNA that you need to keep track of is ribosomal RNA, or rRNA. rRNA is a vital component of ribosomes, the cellular structures where proteins are created. Ribosomes consist of rRNA and proteins, and they provide the necessary framework for protein synthesis to occur. Overall, RNA plays a critical role in the central dogma of molecular biology. It describes how genetic information flows from DNA to RNA to proteins. Without RNA, the process of protein synthesis would not be possible. So that's the key here. RNA is vital to transferring genetic code from the DNA itself, right? The DNA never leaves the nucleus, okay? Other than in replication and cellular division, right? The DNA is like your original. You don't want to ruin your original. You don't want to lose your original. And so you keep your original in place and you require copies to be made for additional procedures, right? So you have your original, that's the DNA. The rRNA comes in, transcribes all of that information that's in the DNA, and then runs it over to the ribosome itself. The ribosome then takes those instructions from the RNA, and that's how protein synthesis occurs. Okay? That's incredibly important to understand that the role that RNA plays in the process of protein synthesis. Until next time. Hey guys, welcome to the lesson on TEAS Bio regarding protein synthesis. So that means protein creation. Let's go ahead and jump in. So proteins are essential to the proper function and health of our cells, and therefore all living things require proteins. In order to create proteins, two processes are required. First, the first process is called transcription. Transcription is the process of your cell making a copy of your DNA using mRNA. Once that new copy is created, the process of translation occurs. This is the process of the ribosome using that strand of mRNA to create a brand new protein. One easy way to remember which process is which is to think about the words themselves. So transcription, think about that. Think about transcribing something. When we're transcribing something, we're writing it down, right? You're kind of copying, in fact. You're looking at this and you're writing down exactly what you see. When your DNA is transcribed, a copy is made using the mRNA. Like we discussed in the prior lesson on RNA, that's exactly what's happening here. The original, the DNA, is protected in the nucleus. A runner, aka your RNA, comes in, takes a look at that DNA, transcribes, makes an exact copy of all the instructions, and then takes that information and brings it to the ribosome. The DNA itself never actually leaves the cell. It's the nucleus of the cell. Translation, that's the next word, that's the next process. When something is translated, it's taken from one language and it's put into another one. This is essentially what happens in this step of protein synthesis. The RNA and the ribosomes take the language of the mRNA and translate it into amino acids that are necessary to form proteins. Let's take a look at each process, both transcription and translation, more closely. So transcription takes place in the nucleus. That's where the DNA is, right? The DNA molecule unwinds, it unzips, and it exposes a specific gene that creates the protein that is needed. RNA polymerase, an enzyme, binds to the gene and starts reading the DNA sequence. As the RNA polymerase moves along the DNA strand, it creates a single-stranded molecule called messenger RNA, or mRNA. The mRNA molecule is complementary to the DNA strand, right? So that means it's adding the opposite of what it sees or what it reads. This means that it is used to make a base pairing to create the new RNA strand. When RNA polymerase sees cytosine, it adds guanine. When it sees guanine, it adds cytosine, period. When it sees thymine, it adds adenine. And then the only difference is when it sees adenine itself, it adds uracil. Once the mRNA molecule is complete, it detaches from the DNA template and leaves the nucleus, carrying the genetic instructions to the next step, translation. Translation occurs in the cytoplasm of the cell, where the ribosomes are. The mRNA molecule exits the nucleus and attaches itself to a ribosome in the cytoplasm. The ribosome reads the mRNA sequence 3 bases at a time. 3 bases are known as a codon, and we're going to use that language a little bit later. Each codon codes for a specific amino acid. There are specific codons known as start and stop codons. These codons tell the cell machinery where the beginning of and end of each amino acid chain is for that specific protein. So how do we get from reading to creating a protein? Transfer RNA. So as we mentioned in our prior lesson on tRNA, those tRNA molecules are specialized RNA molecules that bind both to specific amino acids and specific codons on the mRNA. The unique structure of tRNA allows it to bind and carry specific amino acids, while also having specific anticodons that allow it to bind to the corresponding codon on the mRNA. Then it transfers the amino acid to the new chain. As the ribosome moves along the specific mRNA strand, it joins the amino acids carried by the tRNA molecules together in a specific order dictated by the mRNA sequence. And remember that the mRNA sequence is based on a complementary sequence to the DNA that was in the nucleus. This process continues until the ribosome reaches a stop codon on the mRNA, signaling the end of protein synthesis. The newly synthesized protein is then released from the ribosome and can go on to perform its specific function within the cell. Let's delve a little bit more deeply into codons. So now that you understand the basics of how codons work, that they have start and stop codons, that there are anticodons, how they're used to help create proteins, let's focus on some of the common things that you will be required to know on your TEAS science exam in the bio section. First, you'll recall that we covered start and stop codons. These are specific codons that never change across any code. They signal to the ribosome where to start and where to stop creating a protein. The start codon is AUG, adenine, uracil, guanine. Anytime AUG is read, the ribosome knows it's time to start building a protein. The 3 stop codons are UAG, uracil, adenine, guanine, UAA, that's uracil, adenine, adenine, and UGA. No, not the University of Georgia, but uracil, guanine, and adenine. When the ribosome reads one of these codons, it knows that it has reached the end of the amino acid chain, and it should stop making the protein. Go ahead and review the rest of this lesson. All right. Hey guys, it's Morgan here with another lesson here in the TEAS bio course. This one is focusing on inheritance. And so we'll begin kind of with a larger overview and then narrow in on how traits are passed on from parents to offspring. So inheritance is a process by which genetic information is passed down from parents to their offspring through DNA. We all inherit characteristics, genes, remember those, from our biological parents. We know this from our study of the process of meiosis and cellular reproduction. So how does inheritance work? Most of what we understand about inheritance is the result of the research of Gregor Mendel. You may have heard his name in prior biology classes. Gregor Mendel was an Austrian monk who conducted experiments on pea plants in the 19th century. He made important discoveries about how traits are passed down from parents to their offspring. And his work laid sort of the foundation for the modern field of genetics. So a lot of the laws of inheritance are named after him. So what you'll see is that in this lesson, on the TEAS exam, and just in general, the way that we understand some of the foundational principles of inheritance are called Mendel's laws of inheritance. So let's kind of dive into what that means and explore each one of the laws. So Mendel observed that each individual has 2 copies of each gene. Now we call those alleles. He didn't have a name for them at the time. During the formation of a gamete, these alleles separate, with each gamete receiving only 1 copy of each gene. When fertilization occurs, the offspring receives 1 allele from each parent, thus restoring kind of the pair. The allele, or the trait that expresses itself, depends on whether that trait or allele is dominant or recessive. We'll cover this in more detail when we discuss Punnett squares. The second law, so Mendel's second law, is the law of independent assortment. This law states that the inheritance of one gene does not influence the inheritance of another gene related to something else. Mendel found that different traits, such as flower color and seed shape, were inherited independently of one another. The alleles for different genes segregate and assort into gametes independently during meiosis. This means that the inheritance of one trait does not necessarily affect the inheritance of another trait. For instance, eye and hair color are not inherited together. If one of your parents has brown hair and green eyes, it does not mean that you will inherit their trait for brown hair and you will also inherit their trait for green eyes, for example. This is why offspring typically have a combination of characteristics from their parents. Each trait, each individual trait, is passed along independently, and trait dominance is what determines which traits are expressed physically in the offspring. So let's dive in a little bit more into understanding kind of dominance itself. There is a way to predict which traits show up, or ones that we see in offspring. We call that a phenotype. So let's kind of familiarize ourselves with the term phenotype. Its opposing term would be genotype. So a genotype is an organism's genetic makeup, right? That's where we're going to see the traits from both your mom and your dad, from both parents. Your phenotype is, well, which one of those traits is dominant? Which one physically shows up outwardly, right? Like on your body or in how your body functions, right? So like the pH, the phenotype, reminds you that those are the physical traits, right? The things that are actually expressed. We represent an individual's genotype by letters that represent the 2 alleles, right? And so the 2 alleles for the flower colors from like Mendel's pea plant experiment would be represented as a capital P and a lowercase p. The capital P in that genotype represents the dominant allele, while the lowercase p represents the recessive allele. Now, it's important to understand that we cannot necessarily see an organism's genotype, right? The traits that we can observe in an individual are called the phenotype. A phenotype refers to the observable physical characteristics of an individual. It represents the actual expression of the genes and its influence by the genotype. Phenotypes can include traits like eye color, height, flower color, seed shape, many other things. So the question is, how does a genotype affect an organism's phenotype? Well, that's through dominance. So let's review a few more of the terms that are going to be very important to understanding dominant and recessive traits. So a dominant allele, that's the capital letter. The dominant allele is a trait that will be expressed if it is present in a genotype. A recessive allele is a trait that will not be expressed if it is present in a genotype with a dominant allele. The dominant allele will dominate the recessive one. However, if all that are present are 2 recessive alleles, the recessive phenotype will be expressed. The next thing you need to understand is sort of how we refer to this. Like I just mentioned, if there are 2 recessive traits versus 2 dominant traits, or a mix, 1 dominant and 1 recessive, these all have names. Let's go through the first one. The first is homozygous individual. Homozygous refers to a genotype in which an individual has two identical alleles for a specific trait. These alleles can either be dominant or recessive, but homozygous, right, that prefix homo means the same. So that means that each allele is the same as the other. There are two types of homozygous genotypes. There's the homozygous dominant, represented by two capital letters, right? So in the pea plant example, it would be capital P, capital P. That's homozygous. They are the same. Dominant. They are capital letters. In this case, both alleles are dominant. For example, if we consider the gene for colors in the pea plants, right? A genotype of capital P would be homozygous dominant, where both alleles are for purple flowers. The next type of homozygous genotype is homozygous recessive. In that case, both of the letters that are representing the alleles would be lowercase, right? So lowercase p in the example of our pea plants. In that case, the expressed phenotype would be the recessive trait because you have two recessive alleles, and the color there would be white. The next type of individual is a heterozygous individual. Think about that prefix. Hetero means mixed, right? So heterozygous refers to a genotype in which an individual has two different alleles for a specific trait. In other words, the alleles are not identical. The genotype contains both a dominant allele and a recessive allele. Heterozygous alleles are represented by one capital letter and one lowercase letter. In the case of our example with the pea plants, the genotype capital P, lowercase p, would be considered a heterozygous individual. In that case, the dominant trait, the capital P, is paired with the recessive trait, the lowercase p, resulting in a purple flower type. Because again, that capital P is the dominant trait, so it will override the recessive allele for the lowercase p for the white flower. Since the dominant allele will always be the one that is expressed, we can use that information to predict the likelihood of the offspring having specific traits. Let's do some examples together. In this example, we'll use eye color. We'll say capital B is the dominant allele and represents brown eye color. Lowercase b is the recessive allele and represents the recessive color. Let's call that blue. If an individual's genotype is a combination that includes a capital B for brown eyes, that individual will have brown eyes. So capital B, B, and capital B, lowercase b, will both result in an individual having brown eyes. The only way an individual would have the recessive trait, that the blue eyes, is if they have a recessive allele from both parents for eye color. That means that individual would have to have a genotype of lowercase b, to have blue eyes. Luckily, we have a great tool that helps us understand this better. It's called a Punnett square.
Hey guys, it's Morgan here with another TEAS 7 bio lesson for you. This one is on microbes and microorganisms. Let's jump right in. So microbes, also known as microorganisms, these terms are sort of interchangeable and will be used as such in this video lesson, throughout the lesson page, as well as on your exam. They are a diverse group of microscopic living organisms that include bacteria, archaea, fungi, protists, and viruses. Despite their small size, microbes have a significant impact on various aspects of life. We're going to go over some of them today. They can be found in almost every habitat on Earth, from the depths of the oceans to the soil to even within the human body. Microbes play an essential role in ecosystems, such as cycling nutrients through those systems and aiding in the decomposition of dead organisms. While some microbes can cause disease, the majority are harmless and can actually be pretty beneficial. They can aid in digestion and are important to our immune systems. The study of microbes is also known as microbiology. It continues to reveal an impressive diversity and intricate interactions with other organisms and potential applications for microbiology kind of across different fields of study. Although there are many microorganisms that help us out, many other microorganisms play a significant role in causing disease in humans, plants, and animals. These tiny organisms have the ability to invade the body, multiply, and disrupt normal physiological processes, leading to different types of illnesses. So let's start by laying down the groundwork for the basic types of microorganisms that are involved in disease. The first are bacteria. Bacteria are single-celled organisms. Some bacteria can cause diseases in humans, animals, and plants. For example, certain strains of bacteria can lead to infections like strep throat or urinary tract infection. The next type of microorganism you're going to need to be familiar with are fungi. Fungi are eukaryotic organisms that include molds, yeasts, and mushrooms. Fungi thrive in warm, moist environments. Think about baking bread with yeast, for example, right? It requires water that is kind of around 102, 103 degrees Fahrenheit. You put the little yeasties in there and you'll love that. It's like a nice little warm blanket for it. Add some flour and they have something to eat. Fungi are decomposers, which means they gain nutrients by absorbing organic material from the environment around them. Other fungi form symbiotic relationships with plants or parasitic relationships with a host. Thousands of species of fungi are harmful to humans and can cause such diseases as athlete's foot or fungal infections in the lungs. The next type of microorganism are viruses. Viruses are unique, non-cellular entities consisting of genetic material, so DNA or RNA, and they're enclosed in a protein coat. Viruses can cause diseases by invading host cells and using them to reproduce. Well-known viral infections or viral diseases include the common cold, influenza, and COVID-19. Next are protozoa. Protozoa are single-celled eukaryotic microorganisms that live in water and soil. Protozoa make up the largest group of organisms in the entire world, micro or macro. Some protozoa can cause diseases in humans, such as malaria, which is transmitted by mosquito bite. They can also cause dysentery, which is spread through contaminated water or food. Finally, are parasites. Parasites are organisms that live on or inside another organism. That organism is known as the host. They depend on the host for resources and nutrients. Without the host, the parasite dies. Some parasites are microorganisms, such as certain types of protozoa and bacteria, while others can be seen with the naked eye. They cause diseases by feeding on or damaging the host tissues. For example, parasitic worms like flatworms can live in the intestines of a host, and they can thrive there for months, if not years. The development and spread of diseases caused by microorganisms involves a complex interaction between the microorganism, the host, and the environment. Factors such as a weakened immune system, poor hygiene practices, and environmental conditions can all contribute to the spread of disease. Preventative measures such as practicing good personal hygiene, maintaining a clean environment, getting vaccinated, these are all crucial in controlling the spread of disease caused by microorganisms. Understanding the role of microorganisms in disease helps us appreciate the importance of public health initiatives and personal health practices to safeguard against infectious disease.
What's going on, NurseUp? Welcome back to another lesson of chemistry to help you pass the T7 test. I'm Mr. P. I've taught chemistry for 10 years, and I'm here to help you pass the T7 test and teach you all the chemistry you need to know. Welcome to lesson 7. In this lesson, we're going to learn all about solids, liquids, and gases, and plasma. States of matter. All molecules and atoms exist in a particular state of matter. The state that they exist in depends on how much energy they have. The three main ones that you experience in everyday life are solids, liquids, and gases. Concrete, solid, the air we breathe. is a gas. The water that's in your refrigerator is a liquid. Now, you're probably aware that you can change the state of things like water. You take the liquid water from the fridge, put it in the freezer, it becomes a solid. So why does this happen? Well, things change states because they change energy. Solids have very low energy. The molecules technically still have energy, but they move very slowly. They have definite shape, definite volume. You add a little energy to those solids and the molecules move a little more. They now have indefinite volume. That means they get to move around a little bit. They get to take up the shape of whatever they're in. You pour the water into a glass, it takes on the shape of that glass. But they still have that definite shape, whatever they're in. The molecules have more energy. So we're going up the mountain of energy here. Then finally, the gas phase. If you heat that liquid enough, it becomes a gas. Gases have really high energy. They have indefinite shape, indefinite volume. They just fill up their containers until there's no more space. They just spread out because the molecules have so much energy. We can affect something's state of matter by changing its energy. As we add energy to a solid, the molecules move faster and faster until they pull apart enough to become a liquid. And the same thing goes from liquids to gas. Now, in that change, there are two sort of transition zones, from solid to liquid and from liquid to gas, where the molecules actually need like an extra amount of energy to be absorbed to really break those intermolecular forces holding those molecules together. So I like to visualize this with a graph. Instead of imagining phase changes as just going from point A, B, C in a straight line, it's more of you heat until you get to the point where the melting happens. That extra energy is known as the heat of fusion. You have to add that extra little oomph of energy to go from that solid to a liquid. And then you add more energy until the point where it will become a gas, the temperature which becomes a gas, but then you have to add that extra oomph of energy to actually turn it into a gas. And that is called the latent heat of vaporization. Now, there are two other phase changes that aren't as common. You can actually go from a solid straight to a gas, and from a gas straight to a solid. Solid to gas is called sublimation. Gas to solid is called deposition. We see sublimation with dry ice. That's probably the most popular example of this. Carbon dioxide is a solid at about negative 80 degrees Celsius, and so if you take it out of that environment, it actually goes straight into the gas phase. And so it has this really cool effect, as you might have seen maybe at like a science show or something like that, or maybe in a science class, or in this video. And deposition is something that it's a little bit harder to visualize, but sometimes we see this effect when we see frost in the morning. Water vapor in the air, which is all around us all the time, if the temperature is able to get low enough to the point where it's just gonna freeze, it skips the liquid phase and it becomes straight up frost on whatever surface the vapor touches. And so those processes also happen. It all depends on the energy. Now, there is one other way we can change the phase of something that does not involve temperature, or in addition to temperature, and that is pressure. If we look at something called a phase diagram, it shows us the relationship between pressure and temperature and the phases. And that's pretty cool. You can actually change the pressure of something and affect the phase, not just the temperature. You might see the effect of this a little bit when you change a tire in your bike or your car, because there's a rapid pressure change when that happens. And so sometimes we see water condense from right around where the opening of the valve is, where you're putting the air into the tire. And so that's an example of something where like the pressure change is actually what's causing the phase to change. Now, pressure and temperature are related. We'll talk more about that in a later video. It's not that the temperature doesn't change at all, but the thing that we change is the pressure, and that can also change the phase. Now, there are three major phases, but there's one fourth one that we don't see a lot of in our everyday life, but it's called plasma, and that's a pretty cool phase. You can achieve it by putting a grape in the microwave. Don't try that at home. YouTube it. It's pretty cool. When a gas gains enough energy, sometimes its electrons can get ripped off of the atoms and it becomes ionized. That's a very, very simple way of putting it. But the gist is, Plasma is ionized gas. So, let's sum up. We have three major phases: solid, liquid, gas. Solid is low energy, liquid is higher energy, gas is the highest energy. And we can change between those phases by manipulating the energy of those molecules. We learned about the latent heat of fusion, which is the energy needed to go from that solid to liquid once the melting point is reached. We talked about the latent heat of vaporization, which is the same idea but from liquid to gas. We also discussed deposition and sublimation. You can skip the liquid phase altogether. And we also talked about how you can manipulate the pressure in addition to temperature to change the phase of something. All right, y'all. Study hard. You're going to ace this test. What's going on, Nurse Hub? My name is Brandon Piasecki, or Mr. P, as my students call me. I teach middle school science, and I taught chemistry for 10 years. And I love chemistry so much. I studied it in college, and I'm excited to help you pass the chemistry portion of the TEAS 7 test. Today's objective: students will be able to identify and define the basic structures of the atom. And by the end of the lesson, you'll be able to know what these terms mean: proton, neutron, electron, orbits, electron cloud. And you'll also be able to understand the size and scale of an atom. First, let's define what is an atom. So the word atom comes from the Greek word atomos, which means indivisible. Now, ancient Greek philosophers had this idea that if you took something, and you can actually do this, take a piece of paper or anything you can break into little pieces, and if you rip it enough times, eventually you'll get to a piece that is so small that you can't cut it anymore. Now, obviously, with paper, I can get to a point where I can't rip it anymore. Okay, you see that? So that right there is a very small piece. It's very difficult for me to rip it into any smaller pieces. But even in this tiny piece, there are trillions upon trillions upon trillions of atoms. So what even is an atom? It's defined as the smallest unit of matter. Think of the world, the universe, like a Lego set. Each individual Lego piece in that universal Lego set is an atom. Atoms come together and they make up everything around us. We'll get more into how they bond later on. There are three major parts of the atom: the proton, the neutron, and the electron. Let me show you. Well, because we can't just take a microscope and just look at an atom because our eyes are literally made of them, we have come up with models. Scientists do experiments to come up with models to figure out what an atom must look like. And so the gist is you got some positive stuff, some negative stuff, and some neutral stuff. Now, the positive stuff is called a proton. Protons are a positively charged subatomic particle that gives the atom its identity, who it is. That is located in the center of the atom, in what's called the nucleus. Now, the simplest atom is called hydrogen. Okay, so hydrogen will contain one proton and one electron. The hydrogen proton is in the nucleus, and it has one, which means that it's located right here. So that H, that means that the identity of hydrogen is that it has one proton. Now watch, if I add a proton to it, then it becomes helium. And I'll get more into this in a later lesson, but that just shows you that if I add protons, the identity of the atom changes. Once we get beyond hydrogen, we got to add something else to the nucleus to keep it stable, okay? That's called the neutron. Neutrons are neutrally charged subatomic particles that are located in the nucleus at the center of the atom. In the nucleus, we have protons, positively charged particles, and neutrons, neutrally charged particles. The word neutron has the same beginning as the word neutral. That's how I remember what's what. Okay, the last particle I mentioned is electrons. These are super important, as we'll see later on. They are orbiting, as you see in these circles. All you need to know for this lesson is that electrons are negatively charged subatomic particles that move around the nucleus along circular paths called orbitals. Electrons orbit the nucleus going super fast. Think like speed of light fast. They orbit in these orbitals. These are imaginary paths surrounding the nucleus of an atom where electrons travel. The reason why we say imaginary is because electrons move so fast. In a nutshell, we can't tell exactly where or how they orbit at a given time, so we just imagine that they are orbiting in these circles. Now, because the area around the nucleus that the electron contains is, like I said, a little bit difficult to track, sometimes scientists use what's called the cloud to illustrate where an electron might be orbiting. So, if you're asked about the cloud, the orbital cloud or electron cloud, it is defined as the area around the nucleus of an atom that contains electrons speeding around the nucleus. And when I mean speeding, I mean they're going super duper fast. This is an atom. This is a picture, a model, I should say, of the things that make up everything in the universe. So wild. Now, I mentioned this is a model, and models are great because that was visualized, but they also have drawbacks too, such as, in this model, you know, how much space you say between the nucleus and this first orbital of electrons? Maybe like an inch or two or so. Yeah, that's not a very accurate depiction of how much space is between the nucleus and the first electron. In fact, Carl Sagan, the famous scientist, you may have heard of him before, he said atoms are mainly empty space. Matter is composed chiefly of nothing. That's crazy to think about. So in reality, the nucleus is actually very, 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 very small, and the distance between the nucleus and the first electron is absurdly huge. Let me show you with an illustration, because it's very difficult to describe and see on this model. If this is the nucleus, I'm going to show you where the nearest electron would be. Okay, so this is the electron. Is this how far it's going to be from the nucleus? What about this? What about this? It's way farther. We're going to go on a little journey right now. As I'm walking in this clip, check the top right just to see on the map how far away the nucleus and the first electron actually are if it's at this scale. So crazy. This is how far away the first electron would be if the nucleus were at the downtowner and were the size of that tangerine. It'd be about 1 kilometer away. Hopefully this demonstration helped that Carl Sagan quote make more sense. Atoms, and therefore everything, are mostly empty space. So we know what an atom is. We know they're like little Legos. Well, how little are these Legos? The answer is super duper tiny. Like you can't even see them with the microscope tiny. So a nanometer is 1 times 10 to the negative 9th power. Atoms are 1 order of magnitude even smaller. So they're 1 times 10 to the negative 10th power meters wide on average. That's crazy tiny, like ridiculous. Scientists need to use models to even visualize the atom. We use simulations like this to see things that are super small. So check this out. I mean, you zoom in super, super tiny past DNA, zoom in a little more, and you got some big protein molecules, zoom in even more, and there's some molecules, which are groups of atoms, and that right there is the carbon atom. That's just on for a sense of scale, how small it is. So yeah, they're like super duper tiny. And that wraps it up for our lesson about atoms. We've learned that the atom is the smallest building block, the smallest unit of matter that exists in the universe. It's like the Lego pieces that build up everything else. We learned that they're made of 3 things: protons, positively charged particles; neutrons, neutrally charged particles. They both exist in the nucleus. And electrons, negatively charged particles. They orbit the nucleus in orbitals. They orbit so fast and they're so unpredictable that scientists call the orbitals electron clouds. We also learned that there is so much empty space in the atom, it's absurd. If the nucleus is the size of a tangerine, the nearest electron would be 1 kilometer away. I hope this lesson was informative and I hope it was helpful. What's going on, NurseUp? Welcome back to another lesson of chemistry to help you pass the T7 test. I'm Mr. P. I've taught chemistry for 10 years and I'm here to help you pass the T7 test and teach you all the chemistry you need to know. The video is relatively quick. It's going over a concept called isotopes. Isotopes are the same element with different numbers of neutrons. It's like different flavors of Skittles. Same candy, different flavor. Now, the reason why I say that is because isotopes are the same element. By that I mean they have the same exact number of protons. Quick review, the proton does what for the element? If you said gives it its identity, then that is correct. Let's do carbon, for example. Carbon has three major isotopes: carbon-12, carbon-13, and carbon-14. Carbon-12 is the most common by far. And we know this because its mass is 12.01. Why is it 12.01? Like what? Like Mr. P. How can an atom have a mass of 0.01? Like there's no fractions, there's no like 1/100th of a proton or 1/100th of a neutron in there, right? That doesn't make sense. It's because the mass you see on the periodic table is an average of all of the known isotopes that exist. And carbon happens to have three. Because it's an average, the average mass on the periodic table is going to be closest to the most common isotope. So 99-ish% of carbon is carbon-12. Check it out. This pie chart shows all the different magnesium isotopes. There's magnesium 24, 25, and 26. The average of all of these in terms of abundance and how much exists is 24.31. And that's the mass we see on the periodic table. So I got 4 questions for how many neutrons will each of the following isotopes have? And the way we write isotopes, we write the name of the element like carbon and then a number. The number is the mass. So carbon-13 means carbon with a mass of 13, the isotope with a mass of 13. So if we want to figure out how many neutrons it has, then we just go to the periodic table and we check out carbon. There it is. And we know the mass of an atom is the total number of protons and the number of neutrons. If we have 2 out of those 3 things, we can find the missing piece. So I just take the mass, 13, and I subtract what I know about carbon from the periodic table. Its number of protons is always 6. So I will write 6 protons. And then that equals 7 neutrons. And this makes sense because 6 protons plus 7 neutrons will add up to that mass of 13. Try magnesium 24, see if you can solve that. All right, so what you should do is take 24, the mass, subtract the number of protons of magnesium. If I find magnesium on the periodic table, Mg, where's it at? Where's it at? Remember, that's an alkaline earth metal. It's over here on the left-hand side. Its number of protons is 12. So I will subtract 24 minus 12, and that equals 12. Magnesium 24 has 12 neutrons. Same situation for chlorine 37. Try that one. Chlorine is 17 on the periodic table. So we take 37 minus 17, 20 neutrons. Sometimes you might see an isotope written like this, so like a superscript. And no need to fret, that number is just the mass, written in a different way. It's a little more concise. So, and it's more of a symbolic notation. Sometimes it's called nuclear notation, nuclear symbol notation. So iron, Fe, is number 26 on the periodic table. So if I want to figure out the number of neutrons in this isotope, iron 56, I look for it on the periodic table and I see it's number 26. The proton is 26. So I say 56 minus 26, and that equals 30. All right, after today, you should be able to define isotope. It is the same element with different numbers of neutrons. And you should be able to figure out how many neutrons an isotope has. Okay? We do that by subtracting the mass minus the number of protons. Okay? And there should be an understanding that on the periodic table, the mass is an average of the known isotopes that exist. I hope this helped you understand a little bit more to help you pass that T7 test. You got this. What's going on, NurseUp? Welcome back to another lesson of chemistry to help you pass the T7 test. I'm Mr. P. I've taught chemistry for 10 years, and I'm here to help you pass the T7 test and teach you all the chemistry you need to know. So far, we've learned about atoms, learned about ions and the periodic table. Today we're going to pick up where we left off on the periodic table and teach you some trends and how it's the gift that keeps on giving. It's an amazing resource. And if you know how to use the periodic table, then it can serve you in so many ways. Organization. How can we develop a system of organization that makes it easy to find elements and characterize them by their properties? Now, most of the elements that exist are metals. The most basic organization of the periodic table is that it's got metals on one side and non-metals on the other side. Minus one exception, and that's hydrogen. Some metals are more reactive than others. Obviously, a lot of metals we use every day, like iron and aluminum, don't just react with anything in a crazy explosive way. Over time, they react with oxygen, but that takes a while. So you got groups and periods. Groups are columns, periods are rows. One cool way you can use the periodic table is to understand an element's reactivity. Take group 1, for example, the alkali metals. Super reactive, like ridiculously reactive. Like they don't even exist in their pure states because they'll just react with oxygen in the air. The farther down you go, lithium, sodium, potassium, more reactive you get. Group 2, alkaline earth metals, also very reactive. Not quite as crazy reactive as alkaline metals, but still very reactive. These two groups of metals, very reactive because they have 1 or 2 outer electrons orbiting their nuclei. And it's very easy for them to lose those electrons and give them up to another element. There's a connection between the electrons and reactivity. All in the middle of the periodic table, we have what's called the transition metals. And this is a bit more of a hodgepodge. Many of the everyday metals we use are found here. Also, there are some very rare transition metals we don't see very often. It's a mixture. Also a group called the metalloids. It's kind of a hybrid in this section of the periodic table. It's kind of like they call it the staircase. So it separates the metals from the non-metals, going in that staircase fashion. Silicon's a good example. It's powering the device you're watching this video on. Now, moving on over to the far right-hand side of the periodic table, 2 key groups I'm going to highlight. The halogens, which is a group of non-metal elements. You got gases like fluorine and chlorine at room temperature at least. Now, these are also very reactive. These have almost a full outer shell of electrons. The final group I'm going to highlight is the noble gases. Think of the noble gases as stuck up and not wanting to interact with other elements. Okay, they have nobility. They're like, no, I don't want to deal with all you guys, all the other elements. They have a full outer shell of electrons. They don't have any room. They don't have any room to take any more in and make a connection, make a bond. The periodic table has all these elements organized by these properties. Really reactive metals on the left, really reactive non-metals on the right. On the far, far right, noble gases. Now, I mentioned a phrase throughout this lesson, outer shell or outer ring of electrons, outermost orbit, another way to think about it. Now, there is a magic number that all atoms, for the most part, seek to have. I use the phrase 8 is great to help students remember that 8 is that magic number.
This is known as the octet rule, a super helpful rule. Now there is a trend that goes with this on the periodic table. These groups I'm writing down, 1, 2, 3, 4, 5, 6, 7, and 8 are the number of valence electrons in each of those groups. So if I go to group 1, all the way down that group, that's 1 valence electron. 2, 3, all of those elements. Now, the one exception for this is helium. It only has 2 electrons total, so it does not have 8, but everything else in group 8, aka noble gases, has 8 valence electrons. Now, atoms have 2 options in order to get their full octet, their set of 8 electrons. They can lose or gain. Groups 1, 2, and 3 will lose electrons in order to get their full octet. Groups 5, 6, and 7 will gain electrons in their valence orbital, their outermost shell, to get their full octet. So why does all this matter? Well, if we're trying to make a molecule like oxygen is 2 oxygen atoms. The way that works is because they share to fulfill each other's octet. Oxygen has 6, and if it can share 2 from another oxygen, then it's got 8. 6 plus 2 is 8, oxygen's happy. In a similar way, we can make a molecule like water. Oxygen has 6 valence electrons. Hydrogen has 1. So 6 plus 1 plus 1 more gets you 8. All right, time for a pop quiz. How many valence electrons does sodium have? Check it out. It's in group 1. So if we build up its total number of electrons, the first shell has 2, second shell has 10, the last shell has 1. All right, next one's up. Next up, how many valence electrons are in fluorine? Well, the first orbital has 2 electrons. The second orbital has 7 because it is in group 17, which has 7 valence electrons. This means that fluorine would rather gain an electron than lose an electron. All right, how many valence electrons does magnesium have? Well, it's in group 2, which means that it's got 2 electrons in the first orbital, 8 electrons in the second orbital, and 2 in the valence, the outermost orbital, because group 2 elements have 2 valence electrons. Magnesium would rather lose those 2 electrons to make a bond than gain electrons.
Let's recap. The big idea. The periodic table is a gift that keeps on giving. It has patterns and trends and ways to organize the elements based on their properties. The elements are mostly metals. On the far left, you have the most reactive metals, the alkali metals. Group 1, they have 1 outer electron in their shell. Group 2, alkaline earth metals. Also very reactive, 2 electrons in their outer shell. Big groups for nonmetals, group 17, the halogens, very reactive. And the group 18, the noble gases. All gases, very non-reactive. And the big pattern that I want you to remember is the pattern of group number with outer shell electron amount. So, group 1's got 1 outer shell electron, group 2's got 2 outer shell electron, group 13's got 3, 14's got 4, 15's got 5. Study hard, you got this. Let's pass this test. What's going on, NerdSep? Welcome back to another lesson of chemistry to help you pass the T7 test. I'm Mr. Pete. I've taught chemistry for 10 years, and I'm here to help you pass the T7 test and teach you all the chemistry you need to know. Today, we're learning about chemical bonds. Know your bonds. There are 2 major types, ionic and covalent. I've mentioned ionic bonds before. An ionic bond forms between a metal and a nonmetal. Okay? When you have a nonmetal such as oxygen or a halogen that's very reactive, and they come into contact with a metal, they rip those electrons away from the metal because they're so close to fulfilling their octet. Remember, 8 is great. So if an element like oxygen has 6 valence electrons, it's so close to getting 8 electrons, it's going to just want to take 2 away from another element. Easy examples of ionic bonds. Table salt, sodium chloride. Sodium is a metal, 1 valence electron. Chlorine, 7 valence electrons. All chlorine needs is 1, takes that electron away, boom. That's an ionic bond. So, generally speaking, an ionic bond is when you have a metal combining with a nonmetal, because the nonmetal just takes those electrons away. Now, a second kind of bond, this is more like a potluck. Okay? How many of y'all like potlucks? I love me some potlucks. I'll bring the mac and cheese. That's my favorite, okay? Just saying. A potluck, everyone brings a dish and we share the food. Covalent, okay? Cooperate means come together. Community, sharing. I have 2 young kids. I'm all about sharing. Elements sometimes play nice and like to share. Ionic bonds are like when the older sibling just takes the toy away and doesn't give it back. Covalent bonds are when everyone gets along and shares. So ionic bonds are made between a metal and a nonmetal. Covalent bonds, they happen to be between nonmetals. Prime example, H2O. It's between oxygen, a nonmetal, and hydrogen, another nonmetal. Let's do some examples.
Let's play a little game. Ionic or covalent? Potassium iodide is our first round. Is this ionic or covalent? All right, let's figure it out. First off, how do they bond? Well, we look and we see K only has 1 electron, valence electron, and iodine has 7 valence electrons. So it'll be easier for K just to give that electron away and transfer it over to iodine. Also, K, potassium, is a metal and iodine is a nonmetal. So because of that, this would be an ionic bond. Round 2, Magnesium oxide. Magnesium has 2 valence electrons. Oxygen's got 6. When you add those up, you get 8, so the octet rule is fulfilled. It's easier for magnesium to lose 2 electrons and just transfer them over, and the oxygen's going to gain them. So, magnesium being a metal, oxygen being a nonmetal, this would be ionic. What about high quality H2O? Well, first off, hydrogen is a nonmetal, and so is oxygen. We know this because they're both gases at room temperature. Hydrogen and oxygen are going to want to share electrons in order to fulfill their octet, which they do because hydrogen has 1 electron each. 1 + 1 + 6 equals 8. This is a covalent bond. Electrons are shared. Can you guess what CO2 would be? Carbon dioxide? Again, carbon is a nonmetal along with oxygen. So when we combine those together, it's going to be a covalent bond. They're going to share electrons, fulfill their octet, and be happy.
All right, we reached the end of the lesson, and by this point in time, you should be able to understand and define an ionic bond, a bond where electrons are transferred between 2 elements, and it is usually between a metal and a nonmetal. You should also be able to define covalent bond. It's the potluck. Everyone's happy. Everyone's coming together. Electrons are shared. Usually it's between 2 nonmetals or several nonmetals. Covalent bonds can be huge. Your DNA is held together by covalent bonds. Make sure you practice. Try some of the practice problems to make sure you really got this. Rewatch the video a few times because this is big, okay? This is chemistry. Figuring out how atoms come together and understanding that process. You got this. Let's knock this test out. In this lesson, we're going to talk about how to name compounds. Now, we've already talked about 2 major categories, ionic and covalent. Ionic and covalent are real simple. Ionic is the simplest. Remember that ionic bonds are metals and nonmetals. So naming ionic compounds is really just the name of the metal, just as written. The name of the nonmetal, but then take the end off and add "ide." So, table salt, NaCl, that is sodium, the metal, chlorine is Cl, so we just make it chloride. Sodium chloride, iron chloride, sodium bromide. The list goes on and on. You just literally just add "ide" at the end. That's ionic naming. There is a list of special ions called polyatomic ions. That just means made of many atoms or several atoms. And I'll throw it up on the screen. That list, you gotta memorize them, unfortunately. But those polyatomic ions, that would be the only exception to like the periodic table of nonmetals. But polyatomic ions, they're basically little groups of nonmetals that exist as a group, as like a clique, a friend group of atoms that travel together and bond with a metal. So in naming, that's the only piece that might make it a little bit more complicated than just naming the nonmetal and adding "ide" at the end. An example of some polyatomic ions that you might need to know would be hydroxide, okay, sodium hydroxide, all right, sulfate, something like that. And these, like I said, just memorize those endings, those names of polyatomic ions, you'll be good on that. Covalent bonding. The way we name covalent bonds, we have a set of prefixes. The prefixes are ones that often are familiar to us, like mono, 1, okay? Di, 2, tri, 3, tetra, like the game Tetris, 4, penta, 5, hexa, 6. So these prefixes are similar to prefixes you might recognize from shapes and things that you may have seen in other areas of academics and other classes or everyday life. If you know the prefixes for covalent compounds, the naming process is similar to ionic compounds. You take the first half of the first compound, you take the first half of the first part of the compound, and you count up how many atoms there are. So if you look at the chemical formula and there happen to be 1 carbon, that can be monocarbon or just carbon. The mono is often optional. And then there's 2 oxygens. The prefix for 2 is di. So carbon dioxide. H2O. Dihydrogen, 2 hydrogen, monoxide, 1 oxygen. It sounds a whole lot scarier when you say it like that. It's really just water. Dihydrogen monoxide.
Here's some examples of simple covalent compound naming. CO2, one of the classics. Carbon dioxide. It's called carbon dioxide because it has 2 nonmetals, carbon and oxygen. There are 2 oxygens, so we use the prefix di. This would be carbon tetrachloride. It's got 4 chlorines and 1 carbon. So we say carbon tetrachloride. Similar technique here, sulfur hexafluoride. The biggest thing here is to study the prefixes.
Then as we get bigger and bigger in these covalently bonded compounds, we get into a category of chemistry called organic chemistry. And that word might sound a little scary to you. You may have heard friends who become medical professionals, doctors, nurses go through OChem in college and they say, OChem, organic chemistry, it's super scary and hard. A big reason for that is there's a lot of different variants when it comes to how carbon, which are organic compounds, makes connections. And so there's some systems that we learn as chemists to help us. And one of those is using prefixes. And when we get into organic chemistry, we're talking about chains of carbons. So I'm going to go through like the basics for how to name a carbon chain. All right, so we've done ionic compounds, covalent compounds, like real basic ones. Now we're getting into organic compounds, carbon chains. Okay, naming carbon chains. You got 2 major parts. You have how many carbons are in the chain and how many bonds the carbon is making. Real simply, the prefix would be how many carbons are in that chain. We're only going to go from 1 to 10. Okay, here are the prefixes. All right, as an example, meth, 1 carbon. Eth, 2 carbons. Prop, 3 carbons, and so on. Now, the ending of that compound is going to be how many bonds those carbons are making in that chain. So there's 1, 2, or 3 bonds. If the carbon is single bonded all the way through, it's going to be an ane. Ane ending. Okay? The simplest organic compound, as an example, is methane. You probably heard of it. Methane. It's a greenhouse gas. Cows produce it. Lots of things produce it. But methane is the simplest organic compound. Carbon with 4 hydrogens around it. Now let's make it a little, slightly more complicated. Add 1 more carbon to it. So now there's 2 carbons. Okay? The prefix then changes to eth. If it's 2 single bonded carbons, ethane. And so on. Okay? Now if you got a double bond, it'd be an ene. E-N-E as the ending. And lastly, a triple bond would be ine. There are a few other little pieces to this, and I'll do some examples to show you simple scenarios you might encounter in naming simple carbon chain compounds. Here we go.
It's time for some hydrocarbon naming. Let's start with the simplest. So remember, the prefixes stand for the number of carbons. So if you have a simple compound like CH4, 1 carbon, your prefix is going to be meth. And it's all single bonds, so it's a methane as the ending. Okay, the ending is important too. Let's ramp it up just a little bit. What if we add 3 carbons, all single bonded? Okay, well, we use a prefix for 3. That is prop. This is propane, just like the kind you use on your stove. That's what it is. That's the fuel. So in this example, we have a double bond. Double bonds have a different ending than single bonds. Okay, we still got to check and see how many carbons we have, 2. So I have 2 carbons. They're double bonded, so I'm using the prefix for 2 carbons, eth, and the suffix, the ending for a double bond is ene. So this would be ethene. So to count your carbons, I got 2, check your bonds. You got 2. 2. It's a double bond. Later on, we'll get into kind of why that happens, but this is just for naming purposes right now. And of course, carbon's special. Carbon doesn't just make 1, doesn't just make 2. Carbon can also make 3 bonds. It can make a triple bond. Let's take a look at an example with the triple bond. Again, we're going to count our carbons. There are 3 carbons. Okay, so we know what that prefix is from 2 examples ago. Prop. But instead of propane, this will be propyne. Y-N-E. Propyne. Whenever you have a triple bond, the ending changes to an -ine. So 3 carbons, we have prop. A triple bond, we have the -ine ending. So the name of this compound is propyne.
All right, let's recap this first naming video. We learned how to name ionic compounds, the metal plus non-metal, "ide" at the end. Covalent compounds, you got prefixes. Simple covalent compounds, you always do the prefix first, then the name of the element, and then "ide" at the end. And lastly, simple organic carbon chains. For these, you got your prefixes, the number of carbons in the chain, and then checking the number of bonds the carbon has, making sure you locate where that is on the chain if necessary, and adding the appropriate ending. You got this. What do Kool-Aid and NyQuil have in common? They're both solutions. From sweet summer drinks to life-saving medicine, solutions are everywhere. They're important in many contexts, and today we're going to explore 4 major aspects of solutions that will help you prepare for the TEAS test and your future career. We'll cover water as a solvent, hydrogen bonds, solubility, and molarity. Water is the life-giving molecule, the reason that intelligent life exists as it does on our Earth, and the reason why scientists look for water as a marker for life on other planets. One of the most remarkable features of water is its ability to act as a universal solvent. Its molecular structure with 2 hydrogen atoms bonded to 1 oxygen atom gives water this unique property. You see, water is what we call polar. Polar means that one end of the molecule is more negative than the other, making 2 poles of charge, like the North Pole and the South Pole on Earth. This polarity allows water to dissolve a wide variety of substances. In chemistry, opposites attract, so the negative ends of water will attract positive ends of other molecules, and vice versa, like we see in sugar, for example. This is also why water and other substances like oil don't dissolve, because there isn't that attraction. You know how sugar dissolves so easily in water? That's water's solvent power in action. The water molecules are attracting to the positive and negative ends of the sugar molecules. This also allows our body to absorb nutrients and transport waste. So many benefits. Now, let's talk about hydrogen bonds, the subtle forces that shape water's behavior. Hydrogen bonds occur between the slightly positive hydrogen atoms of one water molecule and the slightly negative oxygen atoms of neighboring water molecules. We see this in a few real-life examples. Have you ever noticed how water forms droplets on like a car windshield or on the surface of leaves? These droplets are due to the cohesive forces of hydrogen bonding. Get it? Due to the... Okay, back on track. This unique property also gives water high surface tension, which allows insects like water striders to glide on water. The concept of solubility is essential in understanding how substances dissolve in a solvent. Solubility refers to the maximum amount of a substance that can dissolve in a given amount of solvent at a specific temperature. Let's imagine this. Trying to dissolve salt in a glass of cold water versus hot water. You know, I think off the bat that more salt's going to dissolve in hot water. This difference in solubility is because temperature affects the rate at which particles move and interact, influencing the dissolving process. Now, how do we measure or make sense of solubility in the lab? There are a few ways, but one of the core methods is measuring concentration of a solution. We use a unit known as molarity to express this. Molarity, often denoted as capital M, is a significant aspect of solution chemistry. It tells us the concentration of a solute in a solution. Specifically, the number of moles of solute per liter of solution. Remember moles from our last chemical reactions video? In order to calculate molarity, we need to know 2 things. the number of moles, and the number of liters. Let's do some example calculations to see how this would look in the lab. All right, let's do this first example. So you have 45 moles and 0.50 liters. Now, we know that molarity is moles over liters. It's literally all we're gonna do right now. It's really nice of this problem. Starting real simple, we're given moles and liters, so we don't have to do anything extra. So, 45 moles on top, abbreviate that MOL, 0.50 liters on the bottom, and I divide these two numbers. So if I have 45 moles and a half liter, that is a concentration of 90, capital M, molar. Very concentrated. Let's do another one. In the lab, usually we measure volume in milliliters and mass in grams. So there are two basic conversions we need to do. We need to be able to go from milliliters to liters and vice versa, and grams to moles and vice versa. In this problem, we're given that the amount of moles in the Kool-Aid is 100.0 moles of Kool-Aid powder. And the volume in this Kool-Aid mixture is 450 milliliters. So we're gonna do the same idea as last time, but we gotta do one extra step. So 100.0 moles, 450.0 milliliters. We gotta turn milliliters into liters. So we know that the prefix milli means 1,000th. So what we can do is say, all right, we have to make sure we move the decimal point 3 places. 1, 2, 3 to the left to show that the amount of liters is going to be basically 1,000 times smaller than this. So we actually have 0.450 liters. So now let's do our division. 100.0 divided by 0.44 divided by 0.450. 222 molarity concentration. So we would say that the answer is 222 molar. Very, very, very concentrated. The idea here is just to practice the process. Okay, so in this example, we are given that we have 21.9 grams of HCl, and we dissolve that in 100.0 milliliters of water. What's the molarity? Well, we do the same exact idea as before. We're gonna find the number of moles over liters. Let's figure this out. And in order to do that, we first need to know how many moles we have and how many liters we have. Once we have that in every problem, once we get moles and liters, we just divide the numbers. So usually the hardest part is getting to those essential units, the mole and the liter. Here we go. 21.9 grams. Let's figure out how many moles that is. We know that if we find the molar mass of HCl, we can use that to find the number of moles, because grams and moles are proportional when it comes to molar mass. So 1 gram of hydrogen is 1.008 grams per mole. And then a gram of chlorine is 35.45. Now this is very nice. We only have one of each. So we're just gonna add up these two numbers. 36.458 grams per mole. I like to set this up in a way that cancels out units I don't need to give me the units that I want. So I usually start off with, when I'm given 21.9 grams, throw it over 1. Now I want to think about this. It's 36.458 grams per 1 mole of HCl. So in other words, we know that 1 mole of HCl equal to 36.458 grams. So I need to figure out what's going to go on top, what's going to go on the bottom. I want grams to go away, leaving me with the moles. It's looking for molarity. Remember? Moles on top, liters on the bottom. So I need to turn this number into moles. So I'll put the mole right there. 1 goes there. And then grams going to go on the bottom. And the cool thing about this, we just cancel out units. Like this. Boom. Boom. Multiply everything to the top. I get 21.9 moles. Dividing that by the molar mass. 0.601. Right around. So I'm going to save that. Okay, it's not the final answer, but I'm going to save it. Now, I want to turn this into liters. I need moles over liters. Remember that if we have milliliters, we can think about milli means 1,000. We know that 1 milliliter is 1/1000th of a liter. So it takes 1,000 milliliters to make 1 liter. The way I can figure out how many liters it is, I'm just going to divide that by 1,000. 1, 2, 3 places on the left. So we have 0.1 liters. We have all the pieces that we need. We're going to take these two numbers and combine them together. Moles first. 0.601 divided by 0.1 is going to be about 6. 6.01 molar is our final answer. Capital M is key. Capital M means molar. One major application of molarity is its use in performing dilutions. Even in the lab and even in medicine, it is important to have the proper concentrations of whatever chemicals you are using. For example, often acids will come in a highly concentrated form and would need to be diluted for most useful purposes in the lab or everyday life. Like vinegar, for example, that's an everyday acid. It's actually 5% acetic acid, and the rest, the 95% that's left over, is all water. We can set up the equation C1 times V1 equals C2 times V2, where C is concentration and V is volume. Often this formula is written as M1 times V1 equals M2 times V2, with the capital M symbolizing molarity, as that's the most common unit of concentration in the chemistry lab. Let's do a few examples to illustrate this idea. All right, the last part of this lesson is going over what we call dilutions. In science, in chemistry, we often need to dilute a sample to get to the concentration that we want. Oftentimes, just in the lab, you have a highly concentrated starting point, kind of like orange juice. You start with concentrate, you add a certain amount of water, it makes nice, delicious orange juice. But the concentrate's like way too much flavor. You gotta dial it down a little bit by diluting it with water. There is a specific equation that we use, C1V1 equals C2V2, or M1V1 equals M2V2 that we're gonna use for this. All right, the equation at the top of the board, so we have that as a reference. Now, you can use a different concentration unit, like percent concentration here. As long as the units are the same, it still works. That's why C1V1 equals C2V2 is also an equation I mentioned. Our problem is telling us that Gabby has 0.500 liters of a 2.5 molar solution. So that's what she has. We're starting there. We are going to write down M1 and V1. So M1 is our starting molarity. Let's make a list. M1, V1, M2, V2. Let's make a big list. M1 is going to be 2.5 molar. It's our starting concentration. The concentration she wants is 1.25 molar. So is that an M2 or a V2? Well, if it's molar, it's going to be M2. So we know she wants to basically cut the concentration in half. 1.25 is half of 2.5. So we are going to put question mark for V2. This will be our variable. All right, let's solve. We'll plug and chug. We're gonna write down our M1 2.5 times our V1 equals 1.25 molar, and then we can just write V2 because that's what we're solving for. Makes it nice and easy. Now, just like in algebra, we're gonna solve for the unknown. So I need to get rid of the other number and move it over so I can divide it out on both sides. 1.25. Out there. 1.25. I like to cancel my units out whenever I can to make sure I end up with the unit that I want. In this case, the unit I want is liters. I want V. I want a volume. Liters is a unit of volume. So I want to end up with that as my unit in my answer. Bring everything down. I can now rewrite this that V2 is going to be equal to 2.5 times 0.5 divided by 1.25, which we can just do all that in the calculator. A little bit of PEMDAS. So 2.5 times 0.5 divided by 1.25. And voila, we get the answer of 1. That's amazing. Nice and easy. So this makes sense. If we want to make it half as concentrated, we would add twice as much liquid, diluting it down, making it more watered down by exactly the proportional amount. In this case, we had half a liter, so if we up it to 1 liter, we're doubling the amount of water, which halves the concentration. Easy way to think about it. All right, the answer is, I would say 1.00 liters. All right, you now have gotten a thorough overview of solutions chemistry. Now you can explain water as a solvent, hydrogen bonds, solubility and how it relates to temperature, and molarity, and even do some molarity calculations. Keep studying hard, and I'll see you in the next video. When I was a little kid, there was this new candy that came out when I was like 10 years old. I remember summer camp at the pool. They were selling these candies called Warheads, and they were like the sourest candy ever. Like crazy, almost painfully sour. Like they were probably just at the borderline of what is legally, you're able to legally put into candy or anything that kids can eat. Now, after about 1 minute of torturous lip puckering and pain in my mouth, they became sweet. That you suck through that, dissolve that layer of sourness. My students still go crazy for sour candies, mega sour candies, and Warheads still exist, and students still love to eat them. They're fun. They're fun. And we know sour flavored foods, we think of them, I think we often will think of them as more acidic, okay? And sour flavored things actually fall into that category of acids. And today we're gonna talk about acids and the opposite of an acid. Anybody know what that is? If you said base, you are correct. We're going to study acids and drop the base, but not really. I'm Mr. P. I've had over 10 years of chemistry teaching experience, and I'm here to help you ace this test. Let's start with the chemistry. Acids and bases are defined as such because of how they interact with one ion, H+. Acids are substances that can donate protons, also known as H+ ions. Think about that for a minute, okay? H+ means it's an H, a hydrogen, that has a positive charge, so it had to lose an electron. Hydrogen only has 1 electron to lose, so if you lose that 1 electron, it literally is just a proton. I went on that quick tangent just because if you hear the word proton in the context of acids and bases, it means H+. So, to sum up, acids are substances that donate protons when they're dissolved in water. This donation of protons results in an increase of the concentration of H+ ions in the solution, which makes it more acidic. More H+, more protons, more acidic. A simple example is hydrochloric acid. When it dissolves in water, it releases lots of H+ ions, leading to an increase in the concentration of H+ ions. Bases, also known as alkaline substances, are the opposite of acids. They are substances that can accept protons or donate hydroxide ions when dissolved in water. This reduces the concentration of H+ ions in solution, which makes it more basic. An example of this is sodium hydroxide. When you dissolve sodium hydroxide, NaOH, it breaks apart into an Na and an OH ion, which causes a big increase in the concentration of OH ions. You may have heard of certain water advertised as being like alkaline or something. Like it's often used in ads, right? This is not health advice, like just FYI, but it's often used, and you might be familiar with the term alkaline from those types of advertisements. That's why I bring that up. Now, we can measure acidity or basicity using a tool called pH. It is a tool used by scientists, doctors, and nurses too, to determine how acidic or basic a solution is. It's very important for our bodies. So pH is very helpful because it gives us a number, a measurable number that we can use to see, okay, are we in a healthy pH range? Okay. So it's very important. This is very applicable to what you will be doing someday soon when you pass this test and get your first job in the nursing field. I like to think of pH as power of hydrogen. It's a logarithmic scale. and it shows us how much H plus ions are dissolved in the solution. And because of its logarithmic nature, the lower the pH, the more acidic a solution is, and the higher the pH, the more basic a solution is. It ranges from 0 to 14, where the lower values indicate more acidity and the higher values indicate more basicity. 7 is considered neutral. Water is neutral. And this would make sense. If you think about what is the ion that is the ion used to measure how basic something is, it's OH, right? What's the ion that's used to measure how acidic something is? It's H+. If you combine OH and H+, you get water. Neutral.